Welcome to the deep dive. Today we're looking into something pretty fascinating, a sort of hidden battleground in cancer. What happens when your immune system encounters a tumor that's, well, basically just starting out, way before it's detectable?
Exactly. It's this whole area called cancer immunoediting. We generally know T cells are key players, but figuring out how they work when the tumor is essentially invisible, that's been the
All
challenge.
right, because how do you study something you can't see?
Precisely. But the study we're diving into today found a really ingenious way. They used a specialized mouse model and some clever molecular tools to, well, make this pre-emergent stage visible.
Okay, let's get into that. The model first, it's the KP-FRT sarcoma model. Why that one specifically?
It's reliable. These mice develop sarcomas relatively quickly and consistently after initiation, so it gives you a predictable starting point, which is, crucial for timing experiments.
And the clever tool.
This is the neat part. They used a fluorescent neoantigen construct. Think of it like installing a tiny bright light, a specific antigen called GP3366, tagged with a red fluorescent protein, M-sherry, which they call MCL-GP3366.
So it glows.
Basically, yes.
Huh.
And because they delivered it using a lentivirus when inducing the tumor, every single cell that starts the tumor journey begins with this bright, I'm here, and I have this foreign antigen signal.
Okay, so you can track the cells, and you know they start out as visible to the immune system because of that neoantigen.
Exactly. It lets you see cell by cell if and when that signal gets turned off if the tumor cell edits itself to hide.
And what was the big picture takeaway from tracking these glowing cells?
It was striking. First, T cells flat out prevented about half the sarcomas from ever forming. Just stop them cold.
Wow, 50%.
Yeah. But the really groundbreaking part was how. The T cells weren't just doing one thing. They seemed to use two distinct jobs, two different functions, to clear out not just the glowing cells of the antigen, but also the nearby cells that had already gone dark.
Okay, two distinct functions. We definitely need to unpack that. But let's start with the initial setup, the comparison between mice with T cells and mice without.
Right. So they looked at tumor emergence, they call it penetrance, over about 150 days. First group, T cell deficient mice. These were either RAG knockout mice who genetically can't make T or B cells, or mice where they'd chemically wiped out the T cells.
And in those mice without the T cell security system.
100% penetrance. Every mouse got a sarcoma. And pretty quickly, too. The median time was around 52 days.
Okay, crystal clear. No T cells, tumor always wins and wins fast. Now, the normal mice, the controls with intact T cells.
Big difference. Only 47% of them developed tumors. So T cells prevented emergence in 53% of the cases. And even when tumors did show up, it was much later, around day 80 on average.
And those tumors that managed to slip through in the normal mice, did they look different?
They did. When they took cells from these emergent tumors and grew them, they were almost universally neoantigen-negative. Basically, 0 to 1% still had that fluorescent signal.
So classic immunoediting. To survive T cells, the tumor had to silence or lose the antigen. Makes sense.
It does. But here's where it gets tricky. The paradox. They looked at the tumors that grew in the T-cell deficient mice, too.
The ones with 100% penetrance.
Yeah. And even those tumors often had cells that had lost the neoantigen. Sometimes just a few percent, sometimes almost all of them.
Wait a minute. If there are no T cells, there's no pressure to hide the antigen, right? So why would those cells lose it anyway?
That's the million-dollar question. It suggests that losing or silencing that antigen is just something that happens, spontaneously during tumor evolution, maybe due to genetic instability or epigenetic changes, regardless of immune pressure.
Okay. But if antigen loss just happens anyway, and T cells were only, say, picking off the antigen-positive cells,
then you'd expect the pre-existing antigen-negative cells to just grow out, right? Maybe the tumors would be delayed in the T-cell-sufficient mice, but you should still get close to 100% penetrance eventually.
right? But they didn't. Over half the mice never got a tumor.
Exactly. So the T-cells must be doing more than just passive selection. They must be actively eliminating all the tumor cells, including those silent antigen-negative ones, at least in those 53% of cases. That's the shift from just editing to active eradication.
Okay, so how did they figure out when this eradication happens? If T cells are going to win, when do they deliver the knockout blow?
They needed a way to control the antigen expression precisely in time. So they used an upgrade on their tool, an inducible deletion system.
Meaning they could turn the antigen off whenever they wanted?
Pretty much. They engineered the construct so that giving the mice a drug, tamoxifen, would genetically snip out the neoantigen gene. like hitting a delete button for that neon sign.
Clever. So they could let the tumor start, let the immune response begin, and then poof, make the antigen disappear at different time points.
Exactly. They tested giving tamoxifen at day 5, day 10, and day 20 after tumor initiation. The idea was if they deleted the antigen before the T cells made their decisive move, the tumor should be rescued and grow out.
And what did that timing reveal?
It was incredibly precise. Giving tamoxifen at day 5 super early deletion rescued tumor emergence significantly. Penetrance jumped back up to 60%, almost like the T-cell deficient mice.
Okay, so deleting the target early saves the tumor. What about day 10 or day 20?
No rescue. If they waited until day 10 or day 20 to give tamoxifen, it didn't matter. The T-cells had already done their work, apparently. Those mice remained largely tumor-free, just like the controls who kept the antigen the whole time.
Wow. So the critical window, the point of no return for the tumor, is sometime before day 10. That's incredibly fast.
It really is. And they backed this up by actually tracking the cells. They looked at the number of transformed tumor cells, the ones carrying the initial viral marker over time.
Yeah.
Those tumor cells peaked around day 6 or 7 and then just plummeted. Disappeared very quickly after that.
And the T cells, when did they show up?
Right alongside that drop.
Yeah.
They measured infiltrating CD4 plus and CD8 plus T cells in the muscle tissue where the sarcoma was initiated.
Yeah.
There was a huge surge, peaking around day 8 to 9, a massive 7.7-fold increase.
So you see the T cells flood in around day 8, and boo, the tumor cells vanish almost immediately after.
That's the picture. The fate of that nascent tumor is basically sealed within the first week or so. It tells us that this immunosurveillance, this pre-emergent editing and elimination, it's happening incredibly rapidly. T cells wipe out most of the danger, both antigen positive and negative, within about 8 days.
Okay, recap time. We've got T cells preventing about half the tumors. They do this extremely quickly, before day 10, and this requires eliminating not just the cells expressing the target antigen, but also the nearby silent ones that have lost it.
Correct. Which brings us back to the how. How do T cells eliminate those antigen-negative bystanders? This is where that dual mechanism comes in.
Right, the two distinct functions, what are they?
Okay, function number 1 is the classic T cell job, IFNγ-independent killing. This is your direct cytotoxic T lymphocyte CTL action. The T cell recognizes its specific target. The neoantigen on the tumor cell engages with it and kills it directly. Standard stuff.
And this doesn't require interferon gamma, IFNO.
Apparently not for the direct kill itself in this context. This direct killing is essential, mind you. It's what puts the brakes on the antigen-positive cells and forces that editing pressure.
Okay, so that handles the antigen-positive cells. What's function number 2, the one that gets the bystanders and explains why 53% of mice get no tumor at all?
That's IFN-dependent killing, also called bystander killing. This mechanism specifically targets the neighboring neoantigen-negative tumor cells.
How does it work? The T cells can't see those cells directly. They don't have the antigen.
They don't need to see them directly. The key is the consequence of the first killing action. When T cells recognize and engage with those few antigen-positive cells, they release a powerful signaling molecule locally, interferon gamma, IFNO.
Ah, okay. So killing the antigen-positive cells is like setting off a flare.
Or maybe calling it an airstrike is a better analogy. The T-cell interaction with the antigen-positive cell triggers this massive local release of IFNO. This IFNO then washes over the entire microenvironment.
And that affects the nearby antigen-negative cells.
Yes. High local concentrations of IFNO can have direct anti-tumor effects on susceptible cells. Even if those cells don't have the antigen the T-cell recognizes, it can slow their proliferation or even induce cell death, essentially creating a hostile zone that kills these bystander cells.
So the few antigen-positive cells act as essential triggers. Their destruction unleashes this IFNO storm that cleans up the neighbors who thought they were hidden.
That's the model. And they tested it directly with an IFNO blockade experiment. They gave mice antibodies that neutralize IFNO.
Okay, so if IFNO is responsible for killing the bystanders, blocking it should save only the bystanders, right? The direct T-cell killing of antigen-positive cells should still happen.
Exactly. And that's precisely what they saw. When they blocked IFNO and looked at day 20, the antigen-positive tumor cells were still gone. T cells eliminated them just fine without IFNO signaling.
But the antigen-negative cells?
They came back. Blocking IFNO restored the number of antigen-negative tumor cells, bringing them back up to the levels you'd see in mice with no T cells at all.
Wow, that's pretty definitive proof. The direct killing is IFNγ-independent, but the crucial elimination of the silent majority, the bystander killing that reduces overall tumor formation, is completely dependent on IFNγ.
It pins down the mechanism beautifully. You need both parts. The direct killing to initiate the response and trigger IFNγ release, and the IFNγ itself to mediate the broader cleanup of the neighborhood.
Let's quickly dive into the micro-level details. They used single-cell RNA sequencing, cRNA-seq, around that critical day 7 time point. What did that show inside the remaining tumor cells?
It really confirmed the IFNγ story at a molecular level. In the tumor cells, they tracked them using a marker called TOM+, that were still around at day 7 in the T-cell sufficient mice. There was a huge enrichment for genes involved in the IFNγ response pathway.
So the cells were clearly sensing and reacting to that IFNγ storm.
Absolutely. And critically, they saw upregulation of a specific gene, CD2H74-4. You probably know it better by the protein it encodes, PDL1.
Ah, the immune checkpoint protein.
Exactly. The tumor cells are trying to protect themselves from the immune attack by putting up PDL1 shields. And that response is driven by the IFNγ they're being exposed to. It's molecular evidence of the battle raging at day 7.
Did the immune pressure affect which tumor cells ultimately survived, if a tumor did eventually form?
Yes, significantly. They used a technique looking at where the initiating virus inserted itself into the genome for basically a genetic fingerprint for each original transformed cell, or clone.
Okay, like lineage tracing.
Sort of. They found that in the T-cell depleted mice, the tumors that emerged were quite diverse, made up of an average of about 5.3 different original clones.
But in the mice with T cells?
Much less diverse. The tumors that escaped were the result of intense selection, averaging only 2.5 distinct clones.
That's clonal narrowing. The T cells act like a filter, wiping out most of the initial diversity and only letting a couple of really tough clones slip through.
Precisely. It paints a picture of immunoediting as this really aggressive, active process that reshapes the tumor, driven by that dual mechanism, direct killing plus that powerful IFNγ bystander effect.
So the big revelation here is the power of that bystander effect. A few cells expressing an antigen can trigger an immune response that eliminates a much larger population of antigen-negative cells around them.
It really changes how we think about immunosurveillance. It's not just about picking off the bad cells one by one. It's about using those initial targets to sterilize the whole area.
Okay, let's bring this home. What does this mean for you, listening? Well, it suggests some mutations, the ones creating these neoantigens, might be doing double duty. They're not just flags saying, kill me.
They might be what we could call guarding neoantigens. Their presence ensures that even if other cells nearby lose their own antigens or never had strong ones, the T-cell response triggered by the guarding antigen can still eliminate them via that IFNγ collateral damage.
And the timing is just paramount. T cells have to get in fast, peak by day 8 or 9, use direct killing for the triggers, and unleash that IFNγ bystander effect to clear the field, all before day 10.
It's a remarkably rapid and coordinated defense.
Which leads to some really provocative ideas for therapy, doesn't it? Instead of just trying to make T cells kill more tumor cells directly, could we design treatments to specifically enhance this bystander effect?
Imagine maybe you could target just a small fraction of tumor cells to make them super antigenic, just enough to trigger a massive local IFNγ release that then wipes out the bulk of the tumor, even the parts the T cells can't directly see.
A localized scorched earth policy triggered intentionally. That's a fascinating thought.
And it leaves us with a question for you to think about. This whole mechanism hinges on those bystander tumor cells being sensitive to IFNγ early on. We saw it works in this sarcoma model, but would it work the same way in all cancers?
Right. Would nascent melanoma cells or early lung cancer cells be as susceptible to that IFNγ-driven bystander killing? Or does the specific tissue environment and the cancer type itself change how effective this guarding antigen strategy might be? Something to definitely mull over.